Good afternoon. Happy Mother's Day. We're into the second lecture or seminar on this issue of the cell. This is a talk that I want you to relax for, because what I'm going to show you is of such overwhelming complexity that there's no way that you can really absorb it other than to note the complexity, and I want you to be impressed by it. The other thing that I'll show you is the level of activity that so much is going on within this little cell that it's hard to believe the level of activity that's going on. So, there's an incredible amount of life throughout this planet of ours. Most of it we have very little contact with because it exists in the form of bacteria. One-cell creatures so small that we only feel their effects when a whole horde of them attack us and make us ill. It's estimated that there are about 10 to the 29th bacteria. 10 to the 29th, remember that's a 10 with 29 zeros on it, so it's a really very, very big number. And there are about 10 times that number to 100 times that number of viruses out there. Fortunately, only a very small number of them are harmful to us, and some of them are actually essential to our well-being. There are estimated to be about 39 trillion bacteria in each of us. They're kind of small, so their total mass is only about 3 pounds. It's a small fraction of your total weight, though they have about as many cells as you're made up of. One study has estimated that the viruses in the ocean cause a trillion infections every second, killing, so each night about 20% of the bacteria in the ocean get killed. Fortunately, you can imagine they're reproducing at pretty much the same rate, so the number remains stable. Now, as I mentioned last week, fortunately, we're not going to have to deal with the important and complex issue of how life began. We'll assume that every living thing came from a similar living parent and is capable of creating creatures similar to itself by whatever reproductive means their species has assumed. Evolution on Earth has been going on now for about 4 billion years and continually manages to gradually modify living creatures. The first humanoids appeared about 2.5 million years ago, and our particular species, Homo sapien, only showed up 200,000 years ago. So we're really very recent newcomers, and having, as I'm sure we all understand, kind of a big impact on this planet at the moment. Now, as I said, life began about 4 billion years ago, and there seem to be 3 major domains of life forms present today, at least as far as biologists are concerned. There's bacteria and archaea. They're typically just one-celled animal, just one-celled creatures, and have been around and never evolved into the eukaryota that are many-celled creatures, which of course is what we are. And the appearance of animals actually doesn't seem to have shown up until about 500 million years ago. Now, the thing that is really amazing is that all these life forms have a common structure. They use basically the same chemicals. They use the same, if I can use the word coda, to interpret what those, how to interpret messages from those things called nucleotides. And we'll be using a bacteria for our investigations for a little while anyway. This E. coli is one of the best studied one-celled creatures on Earth. Its genome, it's so well studied now that we can artificially produce its genome, and its genome has been artificially produced and actually inserted piece by piece into a living E. coli cell. That happened around 2019. So here let's try to just get some scale now for this thing we're plunging into. As I said, though there was a little debate about it. The smallest living cell is made up of about 300 million atoms, which is really quite a large number and rather surprising on the face of it that it took so many atoms in order to make a living creature. If we now try to get a sense of the scale of the size of things, this is a very scientific kind of unit. It's 10 to the minus 9 meters, known as a nanometer. You're about typically 2 meters in length. So this is a billionth of half of you. So you can imagine that's pretty darn small. An atom is about 2 of those nanometers. If you put together some of those atoms to make a molecule like sodium chloride, it's 7. A protein molecule, which are pretty large molecules, would be about 100 nanometers. A cell is typically dimensions of 20,000 nanometers. A human hair, the thickness of your hair, is about 100,000 nanometers. And you, as I said, are 2 billion of those nanometers. If we look at it in terms of mass, how much mass the thing has, I've shown an atomic nucleus, and we're measuring these things in AMU, atomic mass units. It's 1/12 the mass of a carbon atom. And so a carbon atom's mass in those units is 12. And when you make, so the nucleus is the order of 12, the atom, because the electrons don't weigh much, is still, for our purposes, 12. That sodium chloride molecule is 52.5 of those units. A protein molecule has 50,000 of those units. The cell has 2 times 10 to the 12th. And you are 4 times 10 to the 27th. And I've just reproduced that series of 3 different kinds of things. These bacteria and archaea are single-celled things, and the other units over here are multi-cell. That's basically the things you're most aware of. Plants and animals fit into that. These single-celled things are drawn as you probably saw them, maybe even in your, probably not your high school textbook, but maybe what you saw in college. There was a cell, and I don't think there was DNA in many, but some of you might have been there when DNA was put together, and there it's shown. And here is an RNA polymerizing, opening up the DNA, making RNA and feeding it into ribosomes to make the proteins that the cell needs. These multi-cell creatures, like ourselves, have cells that are somewhat more complex in that we have in that they have a nucleus, and in that nucleus is the DNA. It doesn't necessarily appear as a circular thing at all. Many of these things are so big that we have to splice them into many pieces. But the cell structures are quite similar in many ways. Ribosomes play the same game, ending up translating RNA that's generated from reading the DNA and converting it into the proteins that the cell needs. So what are cells made of? Well, because there's an awful lot of water in a cell, about 65% of it by weight is water. Then carbon is another very major makeup, hydrogen, nitrogen, and then trace elements of other more or less light chemicals. There's nothing there that's heavier than calcium on this list. And here is just a breakdown into a diagram showing just how much of us is water, and this would be the other 30% a variety of chemicals. So we'll be looking now at bacteria. They're the simplest single-cell form of life. It's believed that a similar but simpler structure was the earliest life form. Bacteria are not really all that simple, as you're going to see. With a mass a trillionth of a gram, or about 5 times 10 to the 11th, that is 500 billion atomic mass units. And that corresponds, just to give you a sense, of something like 28 billion water molecules. So that's what is a way of thinking about the mass of a cell. Its density, obviously, is very similar to that of water. That's why you more or less float. And hence, with that mass, you're that fraction of a gram, you're the order of 10 to the minus 12 cubic centimeters is the volume of a cell. The length is about 1/100th of a human hair. And what happens, because these cells are so small, is events take place on time scales that are really short compared to what we're used to. We're kind of 1 second creatures, whereas in a cell, things are happening at the order of a 10,000th of a second. Let's see. The bare minimum for any living cell, based on DNA based, as most everything that we know of is a cell wall. That's a means of defining what the cell is. A means of making proteins. There'll be these things called ribosomes, which, if you remember thinking back to what von Neumann told us, that any creature that was capable of reproducing itself had to be able to do. It had to carry a blueprint of itself. It had to have the capability of reading that blueprint. It had to have the capability of executing the blueprint. And it had to be able to make a copy of the blueprint to pass it on to the next cell. So inside bacterial cells, there's no real motors going on, so the only way things move around is by random diffusion. But because a cell is basically so small, it turns out that a big thing like a protein, just by random diffusion, can sense everything that's present inside a cell within a tenth of a second. So that small size allows things to happen on time scales that are very short. And this is a picture we'll be seeing more of. This is an E. coli cell wall, the DNA inside it, and the cytoplasm, which we tended to think of as kind of simple stuff. But thanks to this man, David Goodsell, he's a biologist, a professor of computational biology at the Scripps Research Institute. He's also a painter, and thank God for him. He is able to paint in watercolor what these cells look like. The things that he does are, to my mind, both beautiful and incredibly informative. They were crucial to giving me a sense of what a crowded and complex place the interior of a cell is, and we're indeed fortunate that we have someone like that, as he can show us things that we will never see for ourselves. He functions like the NASA illustrators in the wonderful pictures they draw of the universe for us, except he's showing us the world of the very small, which we also can't see. Some of the structures he depicts are from models, and of course the colors are for aesthetic and educational values. You would not see such a lovely Technicolor display. So this is a cell, an E. coli. It's a 10,000th of a centimeter long, and here's its cell wall. This is the cell's DNA. Don't worry too much about that. We'll learn about that in subsequent in subsequent lectures. But what he then shows you, and to me was absolutely essential. This square box now is magnified by a factor of 10, and this is what is actually in the interior of that cell. Things that you, so the first thing to just look at is say, oh my God. It is just absolutely amazing that that's what's going on in there. This thing that you see, this incredibly long strand, is the DNA of the E. coli, and it sort of gets all wrapped up in the center of the cell. One of the things to immediately appreciate about it is, it looks really tightly wrapped, and it is, because the information that's contained inside it is something that you want to protect very jealously. That is the genome of the creature, and you need very special chemicals to open it up. Don't worry at all about the details of this. I'll be saying some words that I've learned that you don't have to pick up till subsequent lectures. This is an RNA polymerase. got it right. And what's coming out of it is a messenger RNA. That messenger RNA will be taken in here by a thing called a ribosome, and the ribosome will use the information that it reads off this piece of RNA to create the proteins that the cell needs.
Gerry, what's that thing that looks like a shower head or something? The green thing up on the upper right.
This thing?
Yeah.
Yeah, well that's this thing. I don't know what to call it.
Oh.
It's a cilium.
It's a what?
Cilium.
A cilium.
It helps movement.
Yes.
Oh.
Yeah,
It's
so,
Arms for swimming.
yes, you bet. There's all kinds of things in here to name. Here's a bit of a blow-up of what we were looking at. Just again, here's the DNA, here's the RNA polymerase opening up the DNA, reading from it, and making a strand of messenger RNA that's going into a ribosome that's going to make the proteins that the cell needs. But just this is miraculous. How did such a thing come to be? I guess evolution is just, to me, remarkable. So here, if you want to, this is not an exercise for today's class, is to learn every one of these darn things in here. But in your spare time, should it amuse you, you can use this thing to, for example, see, here's 60 DNA. Okay, great. 38 was a ribosome, that was there. An RNA polymerase, here, that was this 60 right there. The DNA was 59, that's the strands. 61 is this messenger RNA. I think at the moment there's probably that's enough. Other than to appreciate it. Now, what he did was to update this. He made that thing, I think, in 1999, and has updated it. It's not awfully different, but it does show you in finer detail very much the same structure. One of the things that I found fascinating were these critters that he drew down here, and they apparently are, I forget their name, but they're really useful in repairing problems in the DNA. There it is. I just can't help but be blown away. It's probably even more magnificent up there. But the DNA.
A question, Gerry, is all that spaghetti stuff DNA?
Yes, it's
Okay.
it's all one piece, one continuous piece.
Thank you.
It has an origin, and when the creature has to replicate itself, it's got stuff that finds the origin and then opens it up, and you have to obviously do things in a very sequential order here, as you'll learn later on. Sequence is really important. Where particular things occur in this strand are absolutely essential. Let's see. More of the same. Okay. So this really, this part I think is extremely important, and it actually was occasioned by having dinner with Paul, with God, what I keep forgetting his last name. Paul.
Spitzer.
Not Spitzer.
Spitzer, Paul Spitzer, there we are. Yeah, and he mentioned that a friend of his had told him that there was more, that the energy density in the human was greater than the energy density of the sun. And that sounded rather implausible. When I thought about it for a minute, I thought, well, yeah, probably true. Most of the mass in the sun is just used to squeeze things in order to make the energy that the sun radiates. So if I define energy density as the amount of energy that something produces divided by its mass, it turns out that a human is roughly about 10,000 times greater energy density than the sun does. Now, in order that the energy, however, in a cell be useful, it has to really occur in very small portions, because a cell, is in many ways a rather delicate thing, and if you get too much energy in inside it, it's done for. So the energy has to be really portioned into very small bits that the cell can make use of for what the cell needs energy to do. In almost all living creatures, it comes from converting andosine triphosphate, ATP, into andosine diphosphate, ADP. So here's the way this works. ATP is a string of 3 phosphate chemical ensembles, and when you break off this one, you get a small amount of energy. About don't worry too much about the unit, other than to appreciate that it's small. 3/10 of an electron volt is the amount that's related that you get every time you break one of these bonds. So if you have ATP and you encounter water and break off one of those bonds, you get that 3 electron volts, and you're left with ADP, that is just 2 of these phosphates. The thing then comes together and uses whatever energy source that particular cell uses. For example, in ourselves and in animals in general, we depend on glucose as that energy source. A plant directly can use as its energy source the sun, and so it uses photosynthesis. So to give you a number, the oxidation of a single one of those glucose molecules, here's that glucose molecule, you oxidize it with 6, in many steps, with 6 water molecules, 6 oxygen molecules, and end up with 6 carbon dioxide molecules and 6 molecules of water. And you've produced 36 of these adenine triphosphate pairs. Thank you very much. Excuse me. So that conversion cycle is continually going on inside your body. It's using ATP, transforming it to ADP, getting some energy to do what it needs to do, and then revitalizing the ADP back into ATP by whatever kind of energy source it uses. If it's an animal like we are, it comes from glucose. If you're a plant, you get that energy from the sun or from light. Use photosynthesis. Now, what was really surprising to me was when you look at the amount of energy that an E. coli produces. It's an incredibly small amount. It's 10 to the minus 14 watts, 4 times 10 to the minus 14 watts. But you divide it by its mass, and you can see it has more than 1,000,000 times the energy density of the sun. A really surprising thing. But what is equally surprising is when you go back and you say, oh, let's see, it's using 4 times 10 to the minus 14 watts, but it's doing it in these little bits of 0.3 electron volts at a time. So you see that it's having the order of 100,000 of these conversions per second. So if you think there's nothing going on in that cell, you're mightily surprised, and certainly I was astonished by this level of activity. And in some sense it shows you, in a way, the cell really has no control over what the devil's going on in there. There's no way you can keep track of 100,000 of those reactions going on at the same time. So it's chemistry that's going on in the cell, facilitated by the various ingredients that the cell is able to produce, and the amount of activity, the amount of energy available to it that it can convert and use in such a useful way. Are there any questions at this stage? Lester,
You want me to ask a question?
ask me a question.
I should clarify.
Please do.
Misinformation. Those filamentous things on the outside of the bacterium, I refer to them as cilia,
Yeah.
but they're not. They are flagella.
They're
There's a
flagella.
difference that I should not.
So
we
you
don't
were-
want to go into. But,
Okay, yeah,
But they're still used for mobility purposes.
you bet.
I don't have any questions.
Uh-huh.
I could express admiration, but
So
Sure.
phosphorus is only 1% of the cell's chemical makeup.
Yes.
So all of this is occurring in connection with just 1% of the cell. Is that right?
yes.
Well, apparently,
Yeah.
yeah.
The rest of the cell is taking a relaxing.
No, no, that phosphorus thing is working on things in the cell. So whenever the cell, remember when you were showing me a nice little diagram how you, in order to get some chemical reaction to go, you need to give it a push?
Yeah.
Well, that's what these ATPs are doing. They come in, the ATP comes into a chemical reaction, and with water, one of those phosphates things is taken off, and the 0.3 electron volts is enough to get the chemical over the barrier and locked into where it needs to be, particularly in the DNA or RNA chain. Yeah.
Jerry, there was a slide back there where there was 2 columns with percentages of various chemicals.
Yeah.
And it wasn't clear what they were. The two, but I think one was probably in percentages in living matter, and the other one was in
Okay.
the universe altogether, or something like that. It's
You want me to go back?
Quite a ways back, yeah.
It's probably
Yeah,
here.
that's the one.
Okay. So this says 15% proteins.
There are 2 columns. What is of the percentages?
So I took this to mean this is by mass And this is by number.
Oh, by number of atoms?
Number of atoms, yeah,
I see.
yeah. That was largely done just, by force of deduction.
Question, you show a good deal of carbon in living tissue, but earlier you said carbon kind of sinks down with the iron, isn't very abundant. Any idea how they accumulate the carbon?
Any idea how they accumulate? Well, let's see, if you were a plant, you are taking carbon dioxide out of the atmosphere. So
Okay.
I can explain it to you when you get to the point that you have photosynthesis. Before that, I'm not sure. Because I think the first life cell, there we go.
Well, Gerry, the bacteria,
Uh-huh.
I don't know a lot about bacteria, but I can assure you that they live in an environment that's aqueous environment
Yes.
and contains lots of stuff. That's why they love soap. Bacteria like sewers as much as they do. So they get all the nutrients from the environment that's in the water in which they're living. And apropos of that, and Paul's question, the number that seems so small to Paul, percentage, but most of the bacterium is water.
Yes.
And so the solid material, phosphorus, is a pretty substantial percentage. Or, if you exclude water from it, or you include water rather in the calculation
Okay.
of all that. That helpful?
Well, it works
Way
for
back
right
there.
now. It works for right now, but in the very beginning, when these things were getting started, you didn't have all the sewer sludge to draw upon, so that was kind of
Didn't
the
have very
question.
many bacteria either.
Yeah.
But it was certainly an aqueous environment, and they just did what they could with what was available.
Yeah. Hi.
Do you have time for a?
Sure.
I wanted to follow up on Lester's identification of this structure, that
Uh-huh.
a flagellum. This has a really important place in the history of science education.
Uh-huh.
Back in 10 or so years ago, the intelligent design proponents took this to court in I think it was in Pennsylvania, and so the support for teaching evolution in public schools came from scientists who challenged the idea that this flagellum is irreducible in structure. This must have been created intelligently. And the scientists could show genetically and morphologically that the components of this flagellum, this rotating little motor, actually had their origin in other bacterial proteins used to take things in and out of the cell, transport proteins. So these two things you raise, evolution and this esoteric structure flagellum, are really connected directly in how we teach science to young people.
Yeah.
The judge, by the way, completely threw out the case for intelligent design, in part because of the science behind this complex structure, the flagellum.
Yeah, there are sometimes structures that even though we know they exist, it's a little hard to establish how they got there. Yeah, evolution is a wondrous thing. Okay.
I think that's at 52 and a half for NaCl. I don't get it. Should be 58 or so, I think. 58 and a half, 23 and 35.
Yeah. Are you still talking about this one?
No, I'm talking about up top there. 52 and a half for NaCl, atomic mass units, just to the left of, that. I think that number's too low, but it's not important.
Okay. It's not far from there. Okay.
35 and a half is 58 and a half.
So now to just try to get at the elements of a cell in terms of their function. So the cytoplasm, that's that fluid inside the cell. It's the medium in which all the chemical reactions take place. It provides a platform for which other organelles can operate within the cell. All of the functions for cell expansion, growth, and replication are carried out in the cytoplasm. The cell wall, obviously, there just to separate the interior, and in a sense to define the cell, the interior contents of the cell from the exterior. The nucleus, in bacterial cells, there really isn't, in a sense, a true nucleus. You find those in eukaryotic cells. The bacterial thing, this free-floating circular strand of DNA is just in the contained clearly in the cell, in the cytoplasm, and it's contains the blueprint for the cell. The ribosomes are an intercellular structure made both of RNA and protein, and it's the site of protein synthesis in the cell. Actually, the ribosome is the thing the cell itself has to end up making. It reads the sequences from the DNA that the messenger RNA brings to it, and translates that via a genetic code into specified string of amino acids, and that ends up making the proteins that needed to make. Don't worry about that. We'll see that in real detail later. And the cell membrane is the thing that allows stuff to go in and out of the cell. The chemical composition, well, proteins are very, very large molecules made of a chain of amino acids that fold up into a particular shape, and that shape turns out to be really important for how things actually function in a cell. And proteins are the most common chemical type found in a biological cell. Polysaccharides, it's a good way to store energy, as it's a collection of sugars. Lipids and fatty acids they're insoluble in water, and they largely make up the cell membrane and wall. DNA, we'll talk much about that in the next lecture, and I don't want to say any more about it. RNA is related to DNA. It's present in all living cells, and with structural similarities really tightly related to DNA. It's single-stranded, and it makes accessible what is contained and wrapped up in the DNA. And here's the approximate chemical composition of a bacterial cell. Again, water. So this is the percent of by weight, and the number of types of molecules of each molecule. Well, with water, there's only one kind of water. So while it makes up 70% of the total mass of the cell, come down here to large molecules, proteins, nucleic acids, and polysaccharides. There you have 96% by mass of the total mass of the cell. But in this case, there are 3,000 different kinds. And so mercifully we are spared having to learn all 3,000 times. I don't know if any, Lester, does anybody know all 3,000 of those?
Oh, I suppose so, but I don't think I know anybody who knows.
I've only got about 2,999.
So, and you can see there are, there's an incredible amount of diversity in a cell, and how that all got figured out and worked out is, to me, just absolutely amazing. But I hope that you got from what I was talking about a sense of just how complex the simplest cell is. A one-celled critter has that kind of structure going on inside it, and that level of activity. 100,000 of those ATPs to ADPs per second going on in the cell. Good Lord. Good Lord. Okay, I'm done with the seminar. I'd love to hear questions or comments. Yes?
Just for a time scale of evolution, could you give us a rough idea of when life was thought to begin and when we began having multicellular organisms?
Okay. There's, of course, a great deal of variability. The beginning is somewhere between 4 billion and 3.5 billion years ago, depending on what kind of evidence you're going to demand. I think quite likely the observation that earliest life was so like whatever it was around that you could, it's hard to tell the difference. But recognized fossils really become evident around 3.5 billion years ago. Then multicellular life, probably not an unreasonable number, is something like 1 billion years ago. And by 600 million years ago, things were getting complicated enough that you could, really see a real creature. Some of the characters that are from the Cambrian explosion are starting to become evident.
Well, it's fascinating that evolution was doing horrendously complex things at the cellular level before we even had what we think of evolution, and this critter leads on to that critter and so on.
Yeah, I think that's an interesting thing to think about. I mean, remember that somehow the thing has to be alive. And once it's alive, and that happened a really long time ago, it's already got quite a level of complexity. It's not clear to me how much evolution has gone on, say, in an E. coli. They certainly are changing like crazy all the time. I think there are the complete genome of 160,000 E. coli, different kinds of E. coli, with various small genetic changes in them. Lester's looking.
No, go ahead.
Yeah.
I'm a little bit puzzled by a question that's floating around in my brain, and that is the cell wall. It sounds like if you could just have a big heterogeneous soup, that these processes could go on without an actual cell.
Well, there needs to be a thing and other. Right?
Yeah. I don't know.
I think he's saying no.
Yeah.
Yeah.
I can comment on it. But
Please do.
Whether it's up to date, I don't know. Charles may know. The origin of life is presumed to be, this is a subject of great interest, of course,
Sure.
that it occurred by spontaneous creation of certain molecules. In particular, the last I heard was an RNA molecule. Just a molecule, which in itself was self-replicating. Does that sound right to you?
Yes, yes. And it had, in answer to this other part of your question, it had the help of...
Louder, Charles.
The other thing that it had besides self-replication was a lipid droplet. If you look at coffee oil, it's been boiled for a while, and look through a microscope, you see these lipid droplets that are very dynamic. They fuse, they divide, quote, divide, so that the action could have been in a simple lipid droplet, oil of some kind. And I think that may have been the kind of envelope that...
And all of this is speculative.
Oh, absolutely. But it's also very good.
Self-replicating RNA molecules have been discovered. So that, it's not implausible, but we don't really know. We're not getting new life created on this planet, and as far as I'm aware, in other words, if there was something new, it probably would be gobbled up or destroyed right away. In fact, I think one of the most interesting things about DNA and the genetic code is that all creatures on this planet have the same genetic code. And it isn't, as far as I'm aware, the code is not imposed by the structure of, say, the amino acids that they assemble into a protein. That it seems random which of 3 nucleotides of a DNA molecule specify a particular amino acid. That's the way the code is expressed. There doesn't seem to be any structural relationship between the amino acid that's being coded by 3 nucleotides. It seems random. But the genetic code is universal.
Yes.
All organisms, bacteria, humans, viruses, all have the same genetic code.
Yeah.
Which implies, to me at any rate, that we're all descended from one creature. When I say creature, just something that is alive and replicating.
Yeah. So a hand up
That's
there.
true, Charles.
What we don't know is how many other attempts might have been
Yeah.
4 billion years ago. How many
Maybe
other
happening
attempts
all the time.
were being, yes, and where did that part of the bushy tree die off?
Gerry, for us novices, what's the functional relationship between RNA and DNA? Not the composition, but...
Yeah, well, I'll get into it much deeper in the next lecture. But basically, the RNA is an open structure of a single strand that actually can copy the information that's on the DNA and actually make it accessible to ribosomes, but make just a general way to look at it, to make it accessible the information that's on DNA, just because it's so hard to get into the DNA because of its tightly twisted character. So it's a way it's the result of opening up the DNA, reading what's on it, and then transferring that information to whatever chemicals need that information to proceed to put together the proteins.
So it acts as kind of a messenger or translator or something in that?
So there is a messenger, and then you need a very good observation. You then need a translator to translate the message, because the message has to be translated from nucleotides to amino acids. And there's a molecule known as tRNA, transfer RNA, that transfers the information. Yeah.
You mentioned the twisted character of DNA. It reminds me of some people I know, but in the textbooks, during cell division, the DNA appears to be all lined up and splits nicely. Does that really happen?
Well, in the kind of thing I'm talking about,
I'm talking about...
I would guess, I don't know how to answer that in the following sense. When in copying the bacteria DNA, it's just one big piece, and it's opened up, and as it's opened up, it's reassembled until you get two, where there was one whole strand, you have now created two whole strands. In the case of the human chromosome, it's very different. Or animal, multi-creature chromosomes. There, it's not one piece, it's many pieces. They're opened in a variety of places, and we'll presumably discuss that in somewhat more detail when we come to that.
I read something quite a while ago that surprised me, I think is interesting, if so, and that is that there is more percentage difference in the DNA between certain bacteria from one another than there is between a human being and a potato.
Well, that sounds to me like an arithmetic problem that I have not, no, it's quite possible.
Yeah.
It's quite possible.
I assume that was a correct statement
Yes.
that I read.
Yeah.
It wasn't written by some idiot.
Yeah, I'd be loath to answer it without sitting down with a pencil. Yes.
Yeah, we had a guest here a while back who's into medical research of some sort, and kind of an example of the conservation of things. He said cartilage is the same in all species. And since I'm wearing a couple of organic valves, that's kind of important so you don't get any tissue rejection, because a pig cartilage is the same as a human cartilage.
I'm sorry, I'm not, is it a question or a comment?
It's a comment.
Oh. Thank you. Yeah. Well, I think the thing that is interesting and revolutionary about looking at cells the way they're being looked at now is they're being looked at a subcellular level. And the fact that you're actually in modifying genes, presumably in a knowledgeable way, but is, I think, something that hasn't happened before. Okay, thanks very much. Next time is going to be how a cell divides.